Hello. Since we have completed the general account and thallus structure discussion and asexual reproduction discussion in Chlamydomonas algae, let us move on to sexual reproduction mechanism in Chlamydomonas. When we are talking about sexual reproduction, the sexual reproduction can be of three types: isogamous, anisogamous, or oogamous. And one thing you have to keep in mind, the thallus of Chlamydomonas can be homothallic. That is, both type of gametes are produced in the same thallus, or can be heterothallic. That you can see plus and minus type of gametes. That is, two types of strains, two different strains of gametes are coming from different parents. We'll see where this happens. For example, in isogamous condition, we'll see that those gametes which are going to fuse will be coming from the same thallus. Okay. And in the case of oogamous condition, we'll see that both of the gametes are coming from different parents. Even in anisogamous condition, we can see that. We'll see that. Now, isogamous. And in botany, remember examples, because the density of such information in your answer make it more weightage, gives it more weightage than your competitor's answer. So, in Chlamydomonas debarianum species, we can observe isogamy. Okay, what will happen? This was a mature thallus. It will be releasing two gametes. These gametes, though morphologically it is looking similar, but physiologically it will be different. That's why its mechanism is inside gene kind of thing. That means its strain, its reproductive strain will be different. That's why it is having the fusing ability. That means you have to write only one sentence. Though these gametes are morphologically similar, it will be physiologically dissimilar. That's why it is able to fuse. So it will come towards the anterior sides towards each other, then it will be fusing, it will be exchanging, or it will be mixing its protoplast. You can see that, and there will be for a few days it will be like this. It will be after exchanging of this protoplasm, there will be a structure, a zygote, which is quadriflagellated. You can see that the two gametes were having two flagella with themselves. So when they mixed, the structure got created. It was having four flagella. So it's a quadriflagellate structure. It is having two nuclei, two pyrenoids, two chloroplast, everything of these two gametes. Now, after swimming like this for some time, this quadriflagellated zygote will lose its flagella and a proper zygote is created. This zygote will be 2N. We know that it was the result of fusion of two different gametes, two haploid gametes. So it is a diploid zygote. This zygote will be developing a thick wall to its outside. So it will be now called as zygospore. Now this zygospore will undergo reduction, division, or meiosis to release the haploid cells. These haploid daughter cells, this will be maturing into parent thallus. So this happens in Chlamydomonas species like Chlamydomonas debarianum, and is an example for isogamy. Now coming to the notes of isogamy, you can read this, everything I have explained. Now, anisogamy you can observe in Chlamydomonas species like Chlamydomonas browni. Okay, in this you can see that the male and female gametes are coming from two different thalli. Okay, one particular thalli will be acting as male gametangium, and another thalli will be acting as female gametangium. Actually, there is a mismatch in the description of these thalli. Actually, this is male gametangium, this is female gametangium. How I was able to observe that thing? Because in the female gametangium there will be only few spores. Okay, there will be only few spores. The spores are very less in number. You can see that only maximum 2 to 4 gametes, and that will be large size in female gametangium. And in male gametangium you can see up to 8 to 16 microgametes. The large gametes are called macrogametes, that will be seen in female gametangium, and the small gametes which are seen in the male gametangium are called male gametes or microgametes. Now this female gamete and the male gamete will be fusing towards each other at its anterior tip, and it will be forming a quadriflagellated zygote for a few period. It will be exchanging its protoplasm. You can see that a diploid zygote is formed, and this diploid zygote will be now developing a wall around itself to form a zygospore, which will be germinating, and it will be releasing the daughter cells, 4 daughter cells. Now what will be the nature of 4 daughter cells? 2 will be of male type and 2 will be of female thallus type. Fine. So it is a note of this anisogamous reproduction phase. Now coming to the final type of sexual reproduction in Chlamydomonas, that is oogamy. Very important. This is observed in Chlamydomonas species like Chlamydomonas ooganum. What will happen? Very simple. You can see that there are 2 thalli. This is female thalli. A is a female thalli and C is a male thalli. Now what we are seeing is this was actually a normal vegetative thallus, but during sexual reproduction in species like Chlamydomonas ooganum, it will behave, one particular thalli will be behaving as a female thalli. For that it will be losing its flagella and it will be waiting like this for the microgametes or the androzoids to meet it. So for that it will be losing its flagella and it will be waiting for the male gametes to fertilize it, because it will be developing an egg within it. Now what will happen is that a male thalli, another male thalli will be developing antheridium within itself. It will be acting as an antheridia, just like it behaved as a nucleonia. It will be behaving as an antheridium and it will be releasing. It will be divided through its protoplast division. It will be forming gametes within and it will be finally releasing this particular androzoids or male gametes which are biflagellated. This will swim and it will reach the tip of the oogonia, and here you can see that then it will be losing its flagella and it will be releasing its male nuclei towards inside. You can see that the female and male nuclei here. Now it will be fusing and forming a zygote as in our previous discussion. Now the zygote will be developing a thick wall around itself called and it will be then called a zygospore. Now this zygospore will undergo meiotic division and it will be releasing haploid cells. These haploid cells will be, some of these haploid cells will be forming male thalli and some of these will be forming female thalli, and this kind of cycles continues like this. So this is the note of this particular oogamous mode of sexual reproduction in Chlamydomonas. You can see that what happens to the zygote. This thing also I have already explained. The zygote will be serving for a few, it will be lying as such for a few period of time by modifying itself into a zygospore by developing a thick wall towards outside. And one point that I missed is zygote accumulates large amount of oils and starch on its body, and because of that the zygospores are red in color, because in these oils and other depositions there will be presence of a pigment called hematochrome. Because of that the zygospores are red in color. So after the resting period it will be germinating and it will be releasing different types of thalli. So this is all about the reproductive phase or modes of reproduction in Chlamydomonas species. So this is all about Chlamydomonas. In the upcoming series of lectures we will see a different algae. I guess we have completed discussing almost all green algae. Maybe one more will be remaining, I guess. Then after that we'll be moving to other algae. Okay, so green algae almost completed. I think only one is remaining. So we'll meet in the upcoming session. Thank you.